Here we'll look at the class Alpha Proteobacteria and order Rickettsiaceae, where we'll look at the important bacteria in Rickettsiaceae and also Anaplasmataceae. However, we'll start off with the bacteria Brucella that causes brucellosis in the family Brucellaceae, order Rhizobialis, also in the class Alpha Proteobacteria. Brucella forms small coccoid gram-negative rods and are cultured under strict aerobic conditions in a capnate environment on enriched media, example on blood agar. So we have Brucella, the species Brucella abortus, Brucella melitensis, and Brucella suis. So Brucella abortus is found in cattle, Brucella melitensis in goats, and Brucella suis in pigs. So these bacteria form a zoonotic relationship with cattle, goats, and pigs, and then they can be transmitted to humans directly from the animals or from indirectly from food, including milk. So the symptoms include undulating fever or Bang's disease. So these bacteria invade the body through the mucosa of upper intestinal and respiratory tracts or through skin. Because these bacteria can avoid lysis um, and can grow intracellular, this bacteria can evade our immune system by continuing to survive in macrophages, and then is transported to lymph nodes. From there, they can spread via the lymphatic system and even go into the bloodstream to enter specific organs. From these specific organs, liver, spleen, bone marrow, etc., this bacteria enters the bloodstream intermittently, causing typical febrile episodes. So we have episodes of fevers accompanied by chills, which gives the name undulating fever. So the incubation period of 1 to 4 weeks. Brucella melitensis infections have more severe clinical symptoms. So for diagnosis, you are able to isolate the bacteria from blood, and the incubation can be up to 4 weeks, and then you can identify it serologically using polyvalent Brucella antiserum. Therapy is doxycycline and gentamicin administered during the acute phase. So antibiotic regimen is given for 3 to 4 weeks. In the order Rickettsiaceae, we'll be focusing on the bacteria in the family Rickettsiaceae, Coxiellaceae, Ehrlichiaceae that causes certain categories of diseases. Named after Howard Ricketts, a scientist who found their existence and died from typhus fever. So they are small gram-negative coccoid or rod-shaped bacteria, and all are obligate intracellular parasites except Rochalimiae quintana that grows on the outside of the eukaryotic host cell, and they have not yet been cultivated in the absence of host cells and require culture in hen embryo yolk sacs or suitable experimental animals, for example mouse or guinea pig. So they are causative agents for typhus fever, Rocky Mountain spotted fever, Ehrlichiaceae, and Q fever. Characteristics: most are arthropod-borne, arthropod-transmitted diseases, where the arthropods will excrete the microbes with the feces, or where ticks will transmit them with their saliva while sucking blood. So these organisms invade the host through skin injuries. So once they enter the host, they will multiply in the cytoplasm of the host phagocytic cell, because they can evade the host immune system and then continue replicating until host cell bursts. They do not survive long outside hosts and must be continually transmitted from animal to animal. Only Coxiella can undergo aerogenic transmission, that is, through inhalation of contaminated dust. They are suited for intracellular lifestyle. Their genes for independent energy metabolism and structural biosynthesis have been reduced. The difference between Rickettsiaceae and Ehrlichiaceae is that Rickettsiaceae reproduce in the vascular endothelial cells, whereas Ehrlichiaceae reproduce in monocytes or granulocytes of membrane-enclosed cytoplasmic vacuoles. So for Rickettsiaceae and Coxiella, we can divide them according to the diseases they cause, whether typhus, spotted fever, Tsutsugamushi fever, or Q fever. So for typhus group, you have the Rickettsia prowazekii and Rickettsia typhi. So you can see that the vector and the host here are arthropod vectors, whether flea or louse. So the disease epidemic typhus, where you have long incubation, about 10 to 14 days, with high fever and very little lethality as high as 20% if untreated. Disease was a problem during World War One and causes a lot of deaths among the soldiers. We also have the Brucella disease, where the symptoms are milder than epidemic typhus. We also have the murine typhus, where you have symptoms also similar to epidemic typhus but milder. So other than the typhus group, you have the Rocky Mountain spotted fever group, where you have Rickettsiaceae, Rickettsia connori, Rickettsia sibirica, Rickettsia acari. So all this causes maculopapillus exanthum, spotted spots on your skin. Sometimes the spots develop into lesions, necrotic lesions, and there is always a bite locus widening around the location where the arthropod ticks or mites have bitten the host. A shorter incubation period with a long fever, 2 to 3 weeks. So minor categories such as the Tsutsugamushi fever, where the symptoms are similar to epidemic typhus, plus local lesions at the bite locus. Q fever by Coxiellaceae burnetii. This is important because it can also be transmitted by dust. It's a problem among farm workers. So you have an incubation of 2 to 3 weeks with interstitial pneumonia and chronic Q fever. You also have endocarditis, infection of the heart, and poor prognosis. Group is Ehrlichiaceae, where you have bacteria Ehrlichia, Anaplasma, Rickettsiaceae, that causes Ehrlichiosis. The main target of the pathogen are monocytes. Ehrlichiosis are present as mild to occasionally severe mononucleosis-like disease with incubation time of 5 to 10 days. When we have the antibiotic of choice, tetracycline, and cultivation from blood using cell cultures exhibit low sensitivity. So most of this identification and detection is using molecular techniques and also using indirect immunofluorescence for antibody titers. Now we'll look at Wolbachia in the family Anasplasmataceae. It's an intracellular bacteria similar to the other families in Rickettsiaceae that infects mainly arthropod species. Okay. And these bacteria have evolved together with the arthropod species into mutualistic relationships where some hosts cannot survive or reproduce without Wolbachia colonization. And it is reported that 25 to 70% of all insect species are potential hosts. Wolbachia was first identified by Marshall Hurtig and Simeon Berthold Wolbach in 1924, and the type species is Wolbachia pipientis. It's pleomorphic, rod-like gram-negative bacteria, and here the importance of Wolbachia is as its potential use as a biocontrol agent. The point here is Wolbachia are ubiquitous in mature eggs, but not sperms. Therefore, only infected females pass the infection to offspring. Wolbachia is a natural bacterium present in up to 70% of all different species of insects, but it is not found in Aedes aegypti, the primary species responsible for human viruses such as dengue, chikungunya, and Zika virus. So for many years, scientists have been trying to use Wolbachia to potentially control mosquitoes that spread these diseases, because they have found that when you introduce Wolbachia into Aedes aegypti mosquito, Wolbachia actually stops these viruses from growing inside the mosquito and being transmitted to people. So because the viruses cannot replicate, the insects will no longer transmit it to their victims and the disease do not spread. So this is an important discovery that has the potential to transform the fight against life-threatening mosquito-borne viral diseases. So the fact that only infected female mosquitoes will pass the bacteria onto the following generation is based on cytoplasmic incompatibility, where if the female is not infected but the male is, the eggs will not hatch. So if both are infected with Wolbachia, all the progeny will have Wolbachia. But if only the female have Wolbachia but not the male, all the eggs will still have, all the mosquito progenies will still have Wolbachia. Biocontrol of mosquito-borne viral diseases, where Wolbachia-infected females are released into the environment, and after a few generations, all the mosquitoes will have the bacteria. And because the viruses don't grow in Wolbachia-infected mosquitoes, then the chain of infection can be broken. Wolbachia does not occur naturally in Aedes aegypti, so we will have to infect these mosquitoes artificially in the laboratory before releasing them. Research have found that these mosquitoes will continue to pass on the bacterium to their offspring up to 13 generations. And because Wolbachia is safe for humans, other animals, and the environment, this is probably the best approach to controlling mosquito-borne viral diseases. So 2 independent risk assessments have been conducted, both of which gave an overall risk rating of negligible for the release of mosquitoes with Wolbachia. It's a much better option than using genetically modified mosquitoes, because these genetically modified mosquitoes, you have to keep buying them, and you have to keep releasing them after a few generations because they don't survive in the environment. And also, you do not know what kind of horizontal transfer might occur for the genetically modified mosquitoes. Very exciting development in the fight against dengue virus, and there are actual research teams in Faculty of Medicine that are working with researchers from Australia to further develop this and to use it in Malaysia.